Welcome to Science Stuff. Today we are going to learn about cell. The term cell is from the language Latin and from the word cella, which means small room. It is the basic structural, functional, and biological unit of all known living organisms. Cells are the smallest unit of life that can replicate independently and are often called the building blocks of life. The study of cells is called cell biology. Let's know the history of research on cell. From the year 1632 to 1723, Antony van Leeuwenhoek teaches himself to make lenses, constructs basic optical microscopes, and draws protozoa such as water cella from rainwater and bacteria from his own mouth. In the year 1665, Robert Hooke discovers cells in cork. Then, in living plant tissue, using an early compound microscope, he coins the term cell in his book Micrographia. In the year 1839, Theodore Swan and Matthew Jacob Sheldon elucidate the principle that plants and animals are made up of cells, concluding that cells are a common unit of structure and development, and thus founding the cell theory. In the year 1855, Rudolf Virchow states that new cells come from pre-existing cells by cell division. In the year 1859, the belief that life forms can occur spontaneously is contradicted by Louis Pasteur. In the year 1931, Ernst Ruska builds the first transmission electron microscope at the University of Berlin. By 1935, he has built an electron microscope with twice the resolution of light microscope, revealing the previously unresolvable organelles. In the year 1953, Watson and Crick made the first announcement on the double helix structure of DNA on February 28th. In the year 1981, Lynn Margulis published Symbiosis in Cell Evolution, detailing the endosymbiotic theory. Through the efforts of all these scientists, now we are going to learn about cell. Cells consist of a protoplasm enclosed within a membrane, which contains many biomolecules such as proteins and nucleic acids. Organisms can be classified as unicellular or multicellular. While the number of cells in plants and animals varies from species to species, humans contain about 100 trillion cells, which is 10 to the power 14 cells. Most plant and animal cells are visible only through a microscope with dimensions between 1 and 100 micrometers. Do you know the egg is the largest visible cell? The cell was discovered by Robert Hooke in 1665, who named the biological unit for its resemblance to cells inhabited by Christian monks in a monastery. The cell theory, first developed in 1831 by Matthew Jacob Sheldon and Theodore Swan, states that all organisms are composed of one or more cells, and that cells come from pre-existing cells, that vital functions of organisms occur within cells, and that all cells contain the hereditary information necessary for regulating cell functions and for transmitting information to the next generation of cells. Cells emerged on Earth at least 3.5 billion years ago. Through the efforts of these three scientists, the basis, information, and research of cell was established. There are two types of cells: eukaryotes, which contain a nucleus, and prokaryotes, which do not. Prokaryotic cells usually are single-celled organisms, while eukaryotes can be either single-celled or part of multicellular organisms. Prokaryotic cells were the first life form on Earth, as they have signaling and self-sustaining process. They are simpler and smaller than eukaryotic cells and lack membrane-bound organelles such as nucleus. Plants, animals, fungi, slime molds, protozoa, and algae are all eukaryotic. These cells are about 15 times wider than a typical prokaryote and can be much as 1,000 times greater in volume. On the screen, you can observe a eukaryote and prokaryote cell. Let's observe. Eukaryotes contain membrane-enclosed nucleus, mitochondria, nucleus, ribosome, cell membrane. Eukaryotes also contain cell wall in some of them, for example like in plants. In prokaryotes, we can find ribosomes, nucleoid, capsule, flagellum, cell wall, cell membrane. Now let's see the differences between prokaryotes and eukaryotes. Typical organisms: bacteria and archaea for prokaryotes, and protists, fungi, plants, animals for eukaryotes. Typical size: 1 to 5 micrometers for prokaryotes and 10 to 100 micrometers for eukaryotes. Nuclear region, nucleus is in the prokaryotes and two nucleus with double membrane is found in eukaryotes. The DNA is circular in prokaryotes and linear molecules with histone proteins is found in eukaryotes. Coupled in the RNA or protein synthesis is coupled in the cytoplasm in the prokaryotes, and the RNA synthesis in the nucleus protein synthesis in the cytoplasm is found in the eukaryotes. Ribosomes in prokaryotes 50s and 30s, in eukaryotes 60s and 40s. Cytoplasmic structure, very few structures are found in prokaryotes, whereas highly structured by endomembranes and a cytoskeleton is found in eukaryotes. Cell movement, flagella made of flagellin is found in prokaryotes. Flagella and cilia containing microtubules, lamellipodia and filopodia containing actin is found in eukaryotes. Mitochondria not found in prokaryotes, 1 to several thousand are found in eukaryotes. Chloroplasts they are not found in prokaryotes. In algae and plants, which are eukaryotes, the chloroplast is found. The organization usually single-celled. These prokaryotes are always single-celled, and eukaryotes can be single-celled or can be in colonies or can be modified or they can be higher multicellular organisms with specialized cells. Cell division. Binary fission is found in prokaryotes. Mitosis or mitosis can be found in eukaryotes. Chromosomes. Single chromosome is found in prokaryotes. More than one chromosome is found in eukaryotes. Membranes are not found in prokaryotes, whereas membrane-bound organelles are found in eukaryotes. On the screen you can observe that it's a plant cell, typical plant cell. The main parts of typical plant cell are filamentous cytoskeleton, small membranous vessels, ribosomes, nucleus, DNA, rough endoplasmic reticulum, leucoplast, cytoplasm, Golgi vessels, which are also known as Golgi apparatus, cell wall, peroxisome, plasma membrane, mitochondria and vacuole, chloroplast, plasmodetta, smooth endoplasmic reticulum. Now on the screen you can observe a typical animal cell. The parts of typical animal cell. Main parts of typical animal cell are endoplasmic reticulum, rough endoplasmic reticulum, ribosome, smooth endoplasmic reticulum, mitochondrion, cell membrane, cytoplasm, lysosome, centriole, Golgi apparatus, nuclear pore, nucleolus, nuclear membrane which form the nucleus. Now let's understand the part of cell. The nucleus, where the DNA is kept and RNA is transcribed. RNA is moved out of cell nucleus through the nuclear pores. Proteins needed inside the nucleus are transported into the nuclear pores. The nucleolus is usually visible as a dark spot on the nucleus and is location of ribosome formation. Ribosomes are when RNA is translated into protein. The protein is called protein synthesis. Protein synthesis is very important to cells. Therefore, large number of ribosomes are found in cells. Ribosomes float freely in the cytoplasm and also bound to endoplasmic reticulum. Endoplasmic reticulum bound to ribosomes is called rough endoplasmic reticulum because ribosomes and endoplasmic reticulum give it a rough sandpaper-like look. These organelles are very small, made up of 50 proteins and several long RNAs bound together. Ribosomes do not have a membrane. Ribosomes fall into two separate units by not synthesizing protein. The endoplasmic reticulum. The endoplasmic reticulum is a transport system for molecules needed for certain changes in specific destinations. Instead of molecules that float freely in the cytoplasm, there are two types of endoplasmic reticulum, rough and smooth. Rough endoplasmic reticulum has ribosomes attached to it as mentioned before, and smooth endoplasmic reticulum does not. Vacuoles. Vacuoles are single membrane organelles that are essentially part of outside that is located within the cell. The single membrane is known in plant cell as tonoplast. Many organisms will use vacuoles as storage areas. Vessels are much smaller than vacuoles and function in transport within and to outside of cell. The lysosome. The lysosome is the digestive system in the cell. It breaks down molecule in the basic components. Digestive enzymes. This demonstrate one of these reasons for having all parts of cell compartmentalized. The cell couldn't use the destructive enzymes if they went sealed off from the rest of the cell. Do you know that lysosomes are called the suicide bags of cell? The cell membrane functions as semi-permeable membrane barrier, allowing a very few molecules across it. it while fencing the majority of organically produced chemicals inside the cell. Electronic microscope examinations of cell membranes have led to development of the lipid layer model. The most common molecule in the model is phospholipid, which has a polar head and two non-polar tails. These phospholipids are aligned tail to tail, so the non-polar areas form a hydrophobic region between the hydrophilic heads on the inner and outer surfaces of membrane. The cytoplasm was defined earlier as the material between the plasma membrane and nuclear envelope. Fibrous proteins that occur in the cytoplasm, referred to as cytoskeleton, maintain the shape of the cell as well as anchoring organelles, moving the cell and controlling internal movement of structures. Microtubules function in a cell division and serve as a temporary scaffolding for other organelles. Actin filaments and thin threads that function in cell division and cell motility. Intermediate filaments are between the sides of microtubules and actin filaments. Yeast fracturing is able to split the bilayer. The Golgi body changes the molecules and divides them into small membrane or contained sacs called vesicles. These sacs can be sent to various locations in a cell. Golgi complexes are flattened sacs of membrane-bound sacs. They function as a packing plant, modifying vessels from rough endoplasmic reticulum. New membrane material is assembled in the various cisternae of Golgi. Mitochondria. Mitochondria are a part of tissue cells that consists of outer and inner membrane. The mitochondria are the main energy source of the cell. In fact, they are often called the power plants of the body because this is where energy ATP is created. Uncoupled thermosis also occurs in mitochondria. Any of the very thin rod-like or string-like structures that occur in nearly all the cells of animals and plants, and that processes food for energy. Like mitochondria, chloroplasts have their own DNA, termed cpDNA. Chloroplasts of green algae and plants are thought to have originated by endosymbiosis by prokaryotic algae similar to living prochloron. Chloroplasts of red algae are very similar biochemically to cyanobacteria. The cellular processes done by all the cells are growth and metabolism, replication, protein synthesis, movement or motility. These four processes, growth and metabolism, replication and protein synthesis, movement or motility, are very important for a cell to survive. Thank you for watching the video. Like and subscribe. Science stuff. If you want any law or any scientific thing to be explained very clearly, please write in the comments below. Thank you for watching the video.